Hello, everyone, and welcome to the second session of Meet Delic's Mushrooms, a three-part live stream series powered by Mydecine. Today, we're joined by an esteemed panel of industry experts where we'll take a look at the journey to FDA approvals, exploring the cultivation of psychedelic mushrooms, developing novel drugs, and how to scale. My name is Kirby Duncan, and I'm the marketing director here at Delic, and today I will quite literally be your captain guiding us through today's discussion. To kick us off, I'd love to give a shout out to our friends and valued partners at Mydecine Innovations Group, who have partnered with Meet Delic to bring you this panel today. Mydecine Innovations Group is an innovative biotechnology company dedicated to improving mental health by developing first and second generation novel therapeutics through world-class infrastructure and drug development standards. The company's renowned medical and scientific advisory board is progressing a robust R&D pipeline of psychedelic-derived therapeutics, novel compounds, therapies, and controlled drug delivery systems. For more information, head to mydecine.net. Special thanks to everyone who showed up for today's virtual event. As much as we love these virtual gatherings, we're looking forward to getting our community together IRL and are pleased to announce our next in-person event, Meet Delic. Meet Delic is the world's premier psychedelic and wellness edutainment event catering to holistic healers, revolutionary businesses and thought leaders, and curious newcomers. Held in Area 15, an immersive and experiential entertainment complex in the heart of Las Vegas, the exciting two-day event features industry entrepreneurs, psychonauts, and leading voices in research, science, and wellness. Be sure to follow Meet Delic on social media at Meet Delic. We've got the full lineup announcement coming very soon, and you won't want to miss it. As I mentioned before, this is the second of a three-part series, so be sure to tune in for our third and final Mushrooms event on July 16th at 11 a.m. Pacific. And if you're not already signed up, head on over to meetdelic.com/virtual to register, and be sure to watch the playbacks from our previous events, including the first Meet Delic Mushrooms webinar on psychedelic integration. Thanks to our audience who came in today. If you have any questions, be sure to type them in the chat, and we'll get them in the Q&A at the end of the show. And now let's get to our panel of experts. First up, we've got Andrew Macisaac. Andrew is CEO of Applied Pharmaceutical Innovations. API is a world-class research facility operating out of the University of Alberta. With their state-of-the-art equipment and facility, they can handle virtually all levels of drug development, including cultivation, extraction, R&D, drug design, analytics, synthesis, and manufacturing. Andrew has worked with the UBC and the University of Alberta and is currently the Assistant Dean for the Faculty of Pharmacy and Pharmaceutical Sciences. Next, we've got Rob Roscow, CSO and co-founder of Mydecine Innovations Group. As a highly educated geneticist, he has spent his academic and professional career looking for valuable and unique medicinal molecules found in nature. The last two companies that Robert applied his innovations to were Canopy Growth and EBU, where he ran their genetics division. Mr. Roscow has already leveraged an expertise in genomics, evolution, and molecular biology to maximize the industrial production of cannabinoids and their use in the pharmacological context. This work has resulted in multiple patent filings and accolades in publications ranging from Nature to Rolling Stones. Now Mr. Roscow has his sights set focus on the vast healing potential of fungi. And finally, Dr. Rakesh Bhat. Dr. Bhat graduated magna cum laude from Leipzig University with a PhD in bioanalytical chemistry, molecular biology, and biochemistry. Dr. Bhat has spent 2 decades working in translational research, and in his previous role, Dr. Bhat worked under Nobel laureate Michael Houghton. Dr. Bhat has extensive translational project management experience as API's Director of Laboratory Sciences. Once again, thank you all so much for being here, and let's get started.
All right, guys, let's get into the questions. So first off, I want to talk about cultivation. You know, we all have our definition of what that means, but Rakesh, I'd love to hear from you. What does cultivation mean in the laboratory setting? You're on mute.
So we are not directly doing the cultivation in our laboratory. So maybe Rob may be the better person to talk about cultivation.
Sure. So the question of cultivation in a laboratory context is really kind of interesting. And so the species of interest that we're really focused on are the psilocybin producing mushrooms, which actually lend themselves to a wide variety of cultivation scenarios. at Mydecine, what we've done so far is really look at being able to produce at scale and then process in a laboratory setting. But we also view in the long run that there'll be ways of leveraging laboratory scale techniques, laboratory style techniques, et cetera, to really amplify and make more efficient the production of psilocybin over the long run, as well as related compounds.
Yeah, here at API, we have a site that's set up to cultivate material that we're getting from Jamaica and other sources around the world. And so that basically the way that we have things structured here is we're looking at various strains and identifying ones that sort of serve high potential. From that point, the team in charge of cultivation take the samples and provide them to Rakesh and his department's responsible for screening compounds to determine, their psychoactive or other biological effects within the laboratory setting.
Got it. And with over 200 species of psilocybin mushroom, how do you focus on certain species for cultivation and research? And what exactly are you looking for?
So one of the things that we really leverage is an ability to screen compounds. And so we can take in these over 200 species and then really look at their production of the interesting chemistry that's held within. And this is really ultimately what the primary focus is understanding this complex chemistry. And iteratively across those 200 plus species, there's quite a bit of undescribed diversity. So our ability to grow these individually and then screen them for interesting chemistry is really the nuts and bolts of how we're going about this.
Yeah, in many cases, when we're talking about mushrooms that contain psilocybin or other chemicals, there's very scant literature on what's inside of them. So one of the sort of core initial steps that we took as we built out the R&D program with Mydecine was bringing aboard the team that could basically work with a very wide range of various biomass and begin that process. And some compounds come up again and again across different species, some which are not in literature of being in a species at all, we found now. And it's really built a very strong repository of knowledge about what's in these plants. As we continue to do research and development work on these, and we determine some of the potential therapeutic effects of some of the compounds we're seeing, we'll also be focusing on narrowing down on those specific strains and coming up with the best scale cultivation to produce products from the commercial market.
Amazing. And Robert, this may be a good time to mention, I read recently that at Mydecine, there were recent advances in the research where you guys discovered 40 new compounds. Can you share more about this exciting news?
Exactly. So this is what Andrew was just describing, our ability to look at the diversity of these mushrooms that we're getting into the facility and then screen them. So grow them at small scale, put them through chemical analysis and understand what chemistry is actually held within. And interestingly, what we found is that the common perception is that there's relatively little chemical diversity in these species, primarily psilocybin and psilocin, but we found is an incredibly large diversity that we anticipate to continue to expand as we continue this research of psilocybin and psilocybin-like compounds, as well as others, but specifically the tryptamines or the psilocybin-like compounds. There's an incredible amount of diversity that's not really well understood yet. And if we think about the end goal here, which is to create better medicines for better therapy, having access to a diversity of compounds is actually really valuable because they all have slightly different properties. And so being able to pick and choose the one with exactly the correct properties for your need or for your therapeutic goal is really beneficial for everyone, including patients.
Yeah, it's worth also adding too that you're not just sort of screening a random plant to determine unknown chemicals. A lot of these chemicals that are contained within these fungi don't serve a purpose other than interacting with bugs or insects or other sort of plant and animal life. A lot of them are very, difficult for the plant to basically, sorry, for the fungus to basically create, and so they have to serve a biological purpose. So if you're looking at potential new therapies and drugs, it's a lot more likely to find something that's pharmacologically active within one of these mushroom species, but particularly ones that are psychedelic or poisonous, versus picking up a blade of grass and figuring out what's inside of it. So it's something that's really, really interesting from that standpoint, because most work hasn't been done on it, and very little work that's been done on it has been done in an integrated fashion, where you can basically take those initial lead chemicals and then have a team like what Rakesh leads doing the screening, and as things move beyond that, move it into animal models and into clinical trials and beyond. So the integrated fashion that we're approaching this research with is something that will likely bear fruit in a much greater way than sort of an independent biologist who's looking at one sort of aspect of a species for just a pure common interest standpoint.
Amazing. And just with this new discovery and everything that we've learned in the last several years, there's so much more to be discovered. I'd love to learn, like, what does that tell you guys? What's the hope for the future and just the opportunity to discover new compounds and mushrooms?
I think we have a really, excitement, a really strong excitement about how we can actually apply the use of these compounds. The depth of diversity that's really hinted at here with these initial 40 compounds that we're seeing is really, speaking to the fact, like Andrew was saying, that this is a really potent group of species as far as drug discovery work. And so this is really kind of the tip of the iceberg. And I'm really excited to be able to, move this into the cellular screening, which will really highlight where and if we have, candidates for new pharmaceuticals.
Nice. And as we think, in the future, bringing these compounds ready, getting them ready for mass consumption, how do you envision the process of producing these new discoveries at scale?
So this is really the second stage of the research. What we've discussed up to this point is the screening, the survey, the understanding the depth of diversity within the fungi, as well as the specific compounds. But as we move forward beyond the survey stage and understand compound X, Y, or Z is of interest for scaling, the species of interest are actually really well adapted to this. And there's strong examples in other species, other compounds of fungi, where you can use leverage laboratory style techniques to really produce these at scale. And you can see this across production systems for a number of nutraceuticals, as well as gourmet mushrooms currently. And so there's known protocols for cultivating these at scale, and then being able to process the chemicals therein. But it's a second phase beyond the discovery stage.
And worth noting too that the potency of a lot of these compounds makes it fairly small footprint to be able to produce for the early stages of the market. So what we've set up right now with our current work is basically capable to produce for any size of clinical trial. As it goes and as it grows, things will need to be looked at in terms of additional capacity for the commercial market, but in terms of production, it's not a huge barrier from that standpoint with these molecules, because we're talking about very, very minute quantities for an active dose compared to other therapeutics, and especially because this is for the medical market. When it comes to psilocybin, there'll be a lot more of a targeted production.
Sure. And, what I'm hearing, a lot of that goes into isolating these compounds, right? So could you guys speak a little bit more to the process of isolating the compound? What accomplishes it, and what are kind of the most commonly used methods to do so?
So unfortunately, we can't give away Mydecine's intellectual property. I think that's the main thing. There's been a lot of work done on figuring out what compounds are within the mushrooms and getting them out, and that's really some of the special sauce, as it were, because it's not a simple process of doing ethanol or methanol extraction of chemicals. You might be getting some but destroying others, and really the work of figuring out exactly what chemicals are in there and the best way to get each one out is a job that we've got a team of 4 scientists working on right now as they continue along these processes. And then we've got experts working on the characterization of figuring out exactly what the structure is from that standpoint, and taking a look at stuff to basically take what we can do on an analytical side and scale it up for the production of materials for clinical trial.
Right. And as we start to bring these or think about bringing these discoveries to market, there's a lot of talk in the industry about natural versus synthetic compounds. Could you dive into the differences between the two, other than obviously the process of making them? Should consumers expect different experiences on them? Share a little bit more about that.
So the concept of natural versus synthetic as it applies to psilocybin is kind of interesting, because this is a compound that is identical whether it's made by nature or whether it's made by a synthetic reaction, if it's purified. And it's the if it's purified part that's really important. And so in the natural context, when consumed in a whole mushroom or a whole mushroom extract, there's all the rest of this diversity of chemistry that we were just mentioning. And that actually makes the anticipated effect of that combination significantly more complex. And so this is where, for our drug development work, we're first isolating compounds singularly and then asking the question, when and if do we actually make some of these combinations? Because it's a very complex space to work to move into, as far as understanding all the minute differences and interactions, synergies when making combinations. Although there's a long-term sort of interest in whether that will help address medical need. There's definitely experiences and examples from polypharmacology, what's often referred to as an entourage effect, where that does actually provide a lot of value to patients and provide value to therapeutic use, but it's just a more complicated question to ask. So it's the step beyond understanding these in singularity.
Right. Okay. So we've talked about cultivation. We've talked about isolation. What is the next step in the research?
So this is really where Rakesh comes in, and what we've set up is a panel of screening steps that allow us to take these interesting compounds and understand where and if they match known pharmacological properties that we would want to then work up into a drug. And so I'll let Rakesh step in and explain a little bit more about how that's done.
Yeah, so the next step is to figure out what pathways does these compounds interact with. So that means we have to do a full library screening of these compounds to look at different pathways or different molecules with what they interact. So there are different type of compound screening what we will be utilizing to see what pathways, how whether they are agonist, antagonist, inhibitor, accelerator, all those methods to figure out what it does finally. So initially we'll be doing it what we call an in vitro. That means in cell lines. Once we figure out in cell lines what pathways it is targeting, then we can go into the in vivo models in mouse models or in bigger sort of non-human primate models to figure out there how does these chemical compounds react there.
Yeah, and it's worth stating that there's so as this process goes along, developing that niche capability to screen is incredibly important for being able to determine the effect. Because if you can't show whether something has value or not, first in a cell and then into a larger animal, it's not viable for for drug development purposes, because you really need that evidence to move forward to the point where you're in preclinical and then in clinical trials with the substance.
Sure, and that lines up with my next question, which it sounds like is perhaps part of the screening process. How do you match a particular active compound to a specific identifier like PTSD, depression, addiction?
So at this time, we know what pathways are involved in PTSD or with addiction, with other things. We know how brain works in these cases. So we target those pathways in the cellular level and see if we can either stop those cellular pathways or we can enhance them. So once we have done that in the cellular process in vitro models, then we go to the in vivo model and show we can produce an PTSD in a mouse model and show that we can cure it, or we can have a mental problem, a disturbance in a mouse model, and give the drug and show that it can correct the problem in mice model or in humans.
Wow. And that also makes me think, we wanted to discuss novel drug design. What are we trying to achieve here with what you've learned in creating new drugs to bring to market?
So as Rob has mentioned earlier, that both getting a novel drug or a drug from natural sources or making it synthetically doesn't mean that it is going to work. That is why we have to do all this drug discovery. We have to do all this screening and show that it is going to work with humans. Right? So yes, it is going to work in animals, it is going to work in the cell lines, and then how we are going to make it work in the humans. So we have to express the human pathways or the proteins within cell lines, and to show that it is working there, we have to show it has good efficacy, it has good specificity. That means if we are targeting a certain disease, it targets only that disease, nothing else, right? It doesn't have any side effect. So drug discovery is a huge process. It is like sort of, you have to first find the target, what target our drug is targeting. And then we have to select the candidate, that it is our selected candidate is only targeting our target, right? And then do the experiments and show the efficacy and the specificity. And once we have that, then we will go to preclinical and the clinical trials, then file the IND, possibly an IP, and then we have a drug in the market.
Yeah, it's worth stating too that sort of the way that the space is turning out when it comes to psychedelics is these things have been at the fringes since the 1960s, and there's a potential for it's a class of compounds, not psilocybin alone. So just like, there were early antidepressants that, had tons of side effects, were not always the best fit. Same goes with the potential of psychedelics. So what we really hope to find throughout this process is, maybe not the first candidates that are the obvious ones, but the ones that truly have that sort of second generation positive effects to sort of come in behind the first products, which is the natural psilocybin, the pure psilocybin by itself. Did I miss anything on that, Rob?
No, I think the one thing that I'd really, highlight here, the excitement around this ability to do drug discovery, match this with screening, and then apply that to novel drug development is really in the comparison. So we know that psilocybin is, already showing promise across a number of areas of mental illness. But if we look at that, it's not a perfect solution. It has a long activity time that's hard to match with therapy. It can cause anxiety in some patients. It may not be metabolized efficiently, et cetera, et cetera. And so these are very specific things that we can actually look at with screening, we can deal with, and then we can, try to make hypotheses and then test them to see if we can actually address some of these features. And, specifically one of the ones that we're very interested in is controlling the duration of the effect, because this is one of the biggest things that's needed to match it efficiently with therapeutic practice. It's uncommon to have a therapy session that lasts 8 or 12 hours. So.
Right. And, in my work, I get asked, when projects will be launched, and it all depends upon a number of factors. So I can only imagine what that means for you guys. But as a hopeful optimist, what is, if you discover a promising compound and get it to a certain, place in the process on your end, what are you hopeful that it takes, 2 years, 5 years, 10 years to bring a drug to market?
So the process that we're talking about here from inception, from discovering a brand new compound in a fungal source, or a slight modification on that in a novel drug development sort of way, really what we're talking about is the first step in a long process of clinical drug discovery, regulatory approval, et cetera. And so the timeframes that you mentioned are not necessarily out of the ballpark. We're not talking about 10 years, but we're talking somewhere around 5 plus or minus. And the important thing to note, though, is that because there's already a body of evidence around psilocybin, what we'll likely see over that 5 years is a continuing wave of new compounds becoming available to the population as prescription drugs. First with psilocybin, and then with these novel ones, these new products of nature that are just being found coming in afterwards, because it just takes time to have the data catch up and have the regulatory approval catch up. But the promise is definitely there.
No, that makes. Go ahead.
Oh, it's just like that, like that example of antidepressant medication. Like there's been generations, and within each generation, there's different variations as well too that work to varying degrees. The initial tricyclic antidepressants that were then followed by SSRIs, and then within SSRIs, which is a class, there was a huge variety that have come out almost every year, new medications that work in different ways. So this is about a pipeline of potential products that will follow the initial work that happens with the sort of classic psychedelics, and continues to hone and refine what's very much an emerging field that will have a lot of potential.
Sure. And Robert, you mentioned in your research creating an experience that isn't as long, right? I've also read some stuff in the news where they're experimenting with taking the hallucinogenic psychedelic trip part of it out. Are you guys doing anything in that space as well?
Certainly the character of the experience is one of the things that's anticipated to be markedly different compound to compound. And you can see this with the combinatorial binding space. So like what Rakesh was mentioning is we have, assays set up to look in singularity at which receptors these compounds are binding to. But in reality, they're binding to a couple all at the same time. And it's this mixture that causes the nuance of effect. And so I think it's an open question as to whether you can fully remove the psychedelic effect and maintain the therapeutic effect, because they operate at the same receptor based on current data. So it would be hard to remove one and maintain the other. But it's still an open question, because there's more going on than just that one receptor. So this is really where our screening comes into play, because we can look at this again, comparing many compounds one to the next, and understand their binding across those larger sets of receptors.
Right. Before we move on to the FDA and all that comes with that, Robert, I read that Mydecine, there's a new initiative that recently launched where there's some machine learning and AI involved in a drug discovery program at the University of Alberta. Can you share more about that and why that's so important?
Yeah, so like we've been discussing here, there's a lot of combinatorial space. If we're looking at a number of different compounds, a number of receptors, and at the bench top, when we're physically touching drugs to cells or drugs to animals, it's much easier to do this from a bottom-up approach. One thing looked at one time, then pairs, et cetera. But what technologies like AI and machine learning allow us to do is they allow us to really look at a similar survey, but this time modeled in the computer, and actually get significantly further as far as the amount of data that we can collect, hypotheses that we can generate in the same amount of time. And this is actually the perfect marriage in a scientific sense, where we have the real world, benchtop work that can then also inform the computer models. The computer models can be used to generate new ideas about what to do on the benchtop. It can actually run as a nice feedback loop where it becomes much more pointed and much more efficient over time. And so in very short, it's a massive way to increase our screening capability.
Wow. Well, that's really exciting. I'd love to move into the FDA approval process. Can we get, from someone a top line overview of the FDA, the role they play in regulating psychedelics and where we stand today?
Sure. So the FDA is, the head regulatory body within the U.S. for drug development and drug approvals, really, for prescription use as well as over-the-counter medications. They regulate anything that you're prescribed by a doctor, the drugs that are sold out of pharmacies, et cetera. And really what that means for us and what that looks like to us as a company is that we need to design clinical trials and show data that's going to prove to the FDA both safety and efficacy for our indications of interest. So we need to prove that there's no safety risks for the patients over time. This starts with the screening that we were just discussing, because you can also screen for adverse properties of the drugs at the same time. And then the next step beyond that is, once we have a good idea of what indications we're treating with this, and a good example is our PTSD work with psilocybin right now in clinical trial. And so we have to structure our clinical trials to show efficacy, or that our compounds and our treatment with the compounds is actually addressing the indication of interest in a statistically representable way. And then you hear often a number of terms around staging or phases of clinical trials. And this is really a stepwise approach where the scale becomes larger and it makes the data more reliable. And as the end of a stage 3, which is a larger efficacy trial, that's when you'll see approval or not approval for clinical use outside of the trial scope.
Nice. So safety and efficacy are a big thing for the FDA. Is there anything that they look for before approving drugs?
It's really the proving of the safety and efficacy. And if you think about this in a psychedelic context, it's really slightly different than what's been approved in the past. And so we often get questions about, is the FDA accepting of psychedelics? Or is this a headwind against the industry, et cetera? And really what it is, to me as a scientist, is really reassuring is that they're very data based. If you can show scientifically that you have safety and you have efficacy against your indication of interest, that's really the bar that you have to hit. And there's a lot of ways to do that. And so there's really not what we see as a company as a headwind against us. We just have to provide the best science to prove to the regulators that we are doing what we say we are. But there's really no specific, special one hurdle one way or the other.
And what are the implications of psychedelics being a Schedule I drug that you come up against with the FDA?
Makes our regulatory steps slightly more difficult, but because everything that we're doing is medically facing, clinical trial facing, these are all steps that can be addressed. And I'll let Andrew jump in and explain that a little bit more. We've done quite a bit in this avenue as far as importing back and forth psychedelics already.
Yeah, so the biggest challenge actually when it comes to the scheduled status of these drugs isn't so much on the clinical trial phase, because by the time you're in the clinical trial, you need very, very stringent approvals in place, whether it be a scheduled drug or a new cancer drug, you name it. Like, those approvals need to be there regardless. However, in the earlier R&D stage and in the production stage, it's a huge barrier for most groups who don't have access to the licenses that are in place to work with these chemicals. So I don't know the full stats, but our group here in Edmonton is, I think, well, definitely in Canada, the only academic lab with the very specific Health Canada dealers licenses and regulatory approvals attached to a GMP facility that can produce for some of these trials and do work with trials, like Rakesh working with samples that are psychedelic, normally he'd have to approve for exemption from Health Canada and a whole bunch of paperwork that will take years just to undergo, some of his basic work. But because we're in an integrated facility where we have most of these pieces in place, it makes it a lot more simpler from that standpoint. There's still a lot of paperwork involved, but it's not the same as basically having to start from scratch. And any single time you want to get hands on a piece of one of these drugs and begin doing research with them. Also, when it comes to making them and those types of aspects, there's a whole bunch of challenges and burdens that thankfully we don't have to contend with as much. And then in the clinical trial stage, you have to set up import-export permits depending on where you're putting the trials, and there's a bunch of paperwork involved, but a lot of that is also tied into the actual clinical trial application. And part of the understanding, as you're getting a controlled substance, is if it's part of this clinical trial, it's probably the secondary amount of paperwork compared to the actual filing of the clinical trial itself.
Sure. And everything from the FDA and beyond, can you share any more challenges that you often come up against or foresee as you continue to push psychedelics into the mainstream?
I think the biggest challenge at this time for the psychedelics is because it is a natural product and people have unlimited access to everything, right? So at this time, that is one thing why FDA is so hesitant to deal with it, because people have access to it naturally, right? And it has been very tough for different companies to make a product out of it, because if people have access to it, why they will buy it? And to sell it as a regulated product, you need to have an intended use. For example, as Robert has been mentioning, we want to target PTSD, depression, and other things. So people have been testing it out of sort of market or without prescription. So that is a biggest challenge at this time, how we can get it as a regulated substance through FDA approvals.
Yeah, I think to that point, actually, Rakesh does raise one of the biggest challenges, at least from my thing, is there's a perception that if something works anecdotally, it must mean it'll be approved. When it comes to getting a drug on the market, that couldn't be further from the truth. People come up with stories of, oh yeah, I did this and had this effect, and they assume that will be taken seriously as a piece of evidence behind a product, but it's not the case. So for the most part, a lot of folks who have been trying to run with their own products or their own discoveries, come off against a brick wall when they hit the regulators, and the regulators say, that's not worth anything. What we need is a really structured, scientifically rigorous, protocol. And yes, the perception of some of these regulators is a little bit affected by the fact that they know that humans have been consuming these substances for thousands of years, but still, at the end of the day, they look at it's a very bureaucratic process to move forward, and there's no shortcuts when it comes to some of the things that need to be done to satisfy regulators.
The way that we're really kind of addressing this as a company, if we think about, how do we actually structure the clinical trials to reach this bar of proof, reach this, level of certainty to, show this to the regulators, what we've done is really set up a simple comparison. And this is honestly one of the best ways of proving a point that's maybe not as, widely accepted, is you set up the most simple comparison that's really hard to argue against. And so the theme that we've been using in our clinical trials is to pair head to head the standard in therapy for the condition of interest. So say with PTSD, the standard cognitive behavioral therapy that has a known efficacy rate or a known success rate. And then we'll pair that directly with the exact same therapy with psilocybin and look to see if we get a higher rate of success for those patients. And so far the answer has been yes, but it's really in setting it up as a very simple black and white comparison that allows us to be much more sure of our answers and move it from, like Andrew was saying, an anecdotal state of knowledge into much more of a clinical and medically accepted one.
Wow. And then, do you see anything outside of the FDA, big money, big pharma, anything else coming through to kind of present a roadblock into getting these adapted into the mainstream?
I think if anything, we'll see greater acceptance from those groups and a shift of the perception that moves it from a roadblock to more of enabler, acceptor, or, especially in the larger pharmaceutical company context. I think there's already a growing interest among those companies and those sorts of players in the space because they recognize that there's, a solid foundation to this research.
Sure. Well, we are coming up on an hour. I want to move into the Q&A, but before we do, we've covered a lot today from cultivation, isolation, and FDA approvals. So panelists, if there's anything else that you'd like to add before I get into some audience questions. All right. Let me pull them up right here. Here we go. What is your goal in identifying these new compounds? And are these findings to be patented or open sources?
So our goal in discovering new compounds, in generating new compounds, is really to have a broader spectrum of effect to then make drugs from. Like we've been talking about, this having a spectrum of these compounds gives us access to different characters of effect, different durations of time, different levels of potency. There's a lot of nuance among this spectrum. And so having access to that allows better manufacture of drugs for targeted conditions. And so that's really the goal with understanding more of these. And then as a company in a patent sense, we really are not looking to patent products of nature, these sorts of things. But what we are looking at doing is using patents to protect our drug development work because it's very expensive and it's really the only sustainable way into the future to fund this work that is actually rather expensive. And so we view patents as a tool to basically fund better access to patients, but in our specific, purpose in that way.
Okay. And next question from the audience. They mentioned the 40 new compounds you've discovered at Mydecine, but I'd love to hear if there's any one compound you've discovered in recent years that has been particularly exciting and maybe one thing we can take away about that compound.
I would say our excitement is honestly more in the breadth of diversity of these compounds than a single one. And because the majority of what we're talking about here are very, very similar to psilocybin and psilocin, it's really that similarity and the diversity around that similarity that's most exciting because it allows us to design new therapies with a lot more variability, a lot more levers to pull as drug designers than we've had in the past.
All right. And next question. I guess, our guests are looking to know how they can stay in the loop. So are there places they can go to learn about upcoming clinical trials and any other ways to stay in touch with what's going on?
So I'd say that one of the best ways is that we have a number of press releases that are coming out. We really like being vocal about what we do as a company. And so following our news that way is definitely a great way. The website that we mentioned earlier is a great way of following us. And certainly as far as the clinical trials specifically, as these are planned, scheduled, and are being conducted, all of them will be available on clinicaltrials.gov. And so there'll be findable there, but also announced by us as they're coming down the pipeline.
Okay. And is there a newsletter that you guys release those releases in? Perhaps we'll send that out to the audience after the event, I think.
Yes, we do. And we can definitely include a link in that information.
Amazing. All right. Well, I want to thank all of our panelists here today. Thank you so much for this valuable conversation. Thank you to our guests. If you came in late, don't worry, we'll be sending a full playback of this in an email next week. We'll also have playbacks from other events, so be sure to comment, show the love, and like. And we will also be sending out ways to stay in touch for clinical trials with Mydecine and API. Thank you everyone so much for coming today. Have a great Friday, guys. We're out.